So I have the great pleasure of introducing Professor Dr. Christoph Meinel. He is CEO and Scientific Director of the Hasso-Plattner Institute, which is located at Potsdam University here close to Berlin. He chairs the Internet Technologies and Systems Group at the Institute. Professor Meinel's academic research is anchored in binary decision diagrams, which is the theoretical underpinning of Boolean functional data structures. His work also encompasses theory development in computational complexity, which forms the basis for simulation models in complexity theory, and that's something of personal interest to myself. Professor Meinel and his team has ported his theoretical work into such applications as telemedicine, cybersecurity, and internet transaction security, which is quite critical in the area of healthcare, electronic health records, and of course to the semantic web, or Web 3.0 as we know it. Professor Meinel.
Thank you, Phil, for the nice introduction. Thank you to the organizers of the World Health Summit to give us the chance to introduce a new technology that has the potential to really speed up the analysis of genetic and genome data. Before I want to start with the talk, I want to invite Dr. Mathieu Chapranov to come here because we want to run an experiment. An experiment, we assume you have 30 days from now on with us for the results.
Okay, just switch to the other laptop. So what we have seen here, we are now starting one of the processing pipelines, and we want to start sequencing one. And we'll show you at the end of the presentation what are the results. So I only press the processing button here, and now let's start the experiment, and I will hand back to Professor Meinel.
Yeah. And now processing is starting in Potsdam, and after 30 days, that is what nowadays is needed. To perform such computation, we can show you the result. So please switch back my slide. So this was the process button. There is a large competition and a big amount of money that's promised for people, that for researchers, physicians, 10 million US dollar will be the height of the award for the first team to rapidly, accurately, and economically sequence 100 whole human genomes to an unprecedented level of accuracy. This is the famous series of genomics prizes. This is the 10th one. So it seems to be a real interesting problem. And so the talk, the agenda is that I want to start a short view on today's situation. I call it conventional medicine. Please forgive me, I am a computer scientist, so it's not in a wrong context. Then I want to mention the vision of the personalized medicine, which requested this analytic task of genome data. Then a few words about the challenges of genome data analysis, and at the end we want to introduce our high performance in memory, that's a new technology, in memory genome project, which is headed by Mathieu. So in the conventional medicine, you have this tissue probe, and then there is an evaluation, the investigation. And actual numbers say that it is an American Cancer Society survey from this year. It says about 38% of the women will develop cancer, and about 45% of men will develop cancer. So if you look what happens if such a disease is recognized and a therapy is decided, started, typically chemotherapy, and if you investigate the success of this therapy, that's not so good figures. So in this study, they counted that only about 25% of these chemotherapies are successful. And the reason is humans are very difficult, and the decision today cannot really take into account these differences. So this is exactly the point where the personalized medicine and activities in this area start, which says in the personalized medicine, one wants to treat patients specially based on their individual dispositions. And these are genomic, genetic disposition, these are environmental decisions and others. And if you look to this development, it's about last 10 years, there is a very big increase in research activities and also in findings. In the morning there was a session which report of many interesting work in this. This work is enhanced by worldwide distributed research institutes that run such projects, that create and have created large expert databases which are freely available also to other researchers and to hospitals. On the other side, there is a limiting factor. All this expert data are collected in different data formats. So it's not so easy to combine this knowledge and in a concrete case to take this distributed knowledge into account. Often it needs human activities to be able to understand and to compare, to see on what level such things can be compared. Here are a list, only a limited list of international famous research institutions that work on this and that provide expert databases in this genomic information about cancer disease. So in a traditional way, a physician detects a patient is suffering from cancer, and then he makes a decision what kind of therapy is done. The idea of the personalized medicine is that this decision should be based on genetic disposition. And to find out this disposition, one starts with a DNA sequencing to analyze the genetic data, the genome, to find possibly mutations, and then to try the expert databases to see whether this mutation is connected to a certain type of cancer or is adapted to good therapy. Then this is a more biological analysis DNA sequence, and then the information technology analysis starts. The analysis of this data, and this is what I speak about. And on the basis of this analysis, at the end, treatment decisions are taken, and the hope and the vision is that such decisions are much more directed to the patient than in the nowadays framework. DNA sequencing, this is about quantity, about 3.2 billion base pairs that have to be investigated. The amount of data size is 1 to 20 gigabyte. And if you look for the analysis of this genomic data, then one knows about 80 million mutations. One knows 20,000 to 25,000 distinct genes, 50,000 to 300,000 different proteins. So one has to do with data, for example, for such alignment analysis or for such variant analysis in the 5 to 10 gigabyte, and for variants 10 to 100 gigabytes. And if you see the time that's needed, then there is the biological, done with the sequencing machines, the DNA sequencing, and then the time that's needed for analyzing this data. And today it's about 30 days the process is needed, and we want to show our application of the in-memory technologies that was developed in Hasso-Plattner Institute together in strong cooperation with SAP. That was originally not developed for this purpose, but we saw that it's very usable here in this context. So if you look to the analysis task of genomic data, then there are mainly two groups of analysis work. One is to analyze the alignment and the variant calling. And the other is to analyze of annotations you can find in the expert databases that are internationally distributed. If you look to the first group of computations, then these are CPU consuming. So the performance of the CPUs here is the factor that determines the speed of this analysis. Typically, the duration is hours. If you look for this analysis part to find out what is known worldwide in the database about this special mutation, then memory capacity is the bottleneck, is what is needed to perform this analysis in a fast way. And nowadays, you need weeks to check all the expert databases to find out is about the situation you want to analyze some knowledge recorded. With our approach, with this in-memory technology, we can break down the alignment and variant calling processes from hours to seconds to minutes, and more interesting, the analysis of the annotations from weeks to real time. To real time, so that is the vision that this contributes not only for research, the vision is that we can contribute for treatment, for the practical work of physicians. The technology behind this speed up, or what is needed by the technology, or what this technology is based on, is on the one side multi-core. It's a development in hardware that many processors are available, and you can use this in parallel, but the problem is you have to break the computational task into independent pieces. Each piece can be computed by one core, and then you have to think how you can bring together the results of this separated computations. The memory-based analysis of annotations here, this is another trend, an actual trend in hardware, and this is a trend that the main memory is that what's close to the processor, this becomes cheap. In the past, this was a bottleneck. So you need this external memories, and the speed to bring data from the external memory close to the processor in the main memory, which has only limited space, is 100,000 times slower than the computation. So in the moment when this main memory becomes larger, you can put all the data at once in the main memory close to the processor, and then you can perform the computations, which drastically reduces the time you need for such a computation. At the end, it's not only this memory. You have also to learn how to make use of this memory, so database management technologies need to be adapted. But this is what this promise and the possibility of this in-memory technology for speeding up the analysis of genome data is. Mathieu, should we look what meanwhile was computed?
To give you a better understanding about what we are talking about, I'd like to give you a visual feedback. So now back to our initial start. So what you're seeing here is our research prototype. On the left-hand side, you see a list of already computed tasks. You can select here some of your sequenced FASTQ files. You can upload them, or you can individually choose from them. We have selected some of the algorithms we can do in our pipeline and the reference genome. And initially, we started to process them, and we see here are some of the results. And for instance, I click on this result set, and what you see here instantly is the list of variants and possible mutation sites. This is sorted like you know it from Google, because on the first side, you see these mutations which are probably connected to diseases, you know.
So you started with analyzing genomic data that's available, and then you try to analyze what kind of mutation or what kind of things you can detect.
Exactly. This is the task which is typically taking some weeks, because you have to do it manually. You have to see which of the mutations is relevant for your patient. You have to evaluate what additional literature is available on the internet, which diseases are connected or mainly interested for you. And what you're seeing here, this is not sorted the typical way. You see here some additional information like the other syndrome, the names of the chromosomes which are affected. And for instance, if you click on one of the entries, you will navigate directly to a genomic browser, which shows you this selected mutation in more detail. So what you see here on the top side, you see the reference genome and how it looked like with the amino acids and the different reading frames. And below you see the selected cell line. You see there is a single nuclear polymorphism here. The C is mutated to T, for instance. And just when I click here, you have seen that I've parsed some of these databases. So I've done the real-time annotation, and all these informations are instantly grabbed from the in-memory database and associated with that mutation. So for instance, if there is new research results available on the internet and integrated in the in-memory database, you will see them immediately here. So you do not have to do your whole sequencing task. You only have to click here instantly, and you will see, okay, there's some new information available for you. And you see typically links like these huge databases like the DB-SNP, and you see the GGB and the Cosmic Cancer from Sanger. And all these informations are grabbed in that moment you need them. And this is traditionally a task you have to do manually, because you have to search on the internet, you have to investigate these sources, and maybe you are using some internal databases which are synced with them, but they are outaged because they are synced once in a quarter of a month or once in a year or whatever. And therefore, this is a way where you can get additional information very fast. And to give you an impression how fast it is, we can select another mutation here, and you can click on that. You see that's different. You see there is, again, another mutation, and you click here and see instantly the details about that. You see also PubMed articles are connected. And this is only one impression how personalized medicine could look like for, on one hand, the physicians which are working with that, but on the other side also for researchers that need to investigate the source, the roots for some of the common cancer diseases, for instance. Back to you.
Thank you. So this is the amount of data, 45 million reads from 1K genome project, 400K to 700K variants that are detected by different algorithms. So we do not develop new algorithms. We base on the algorithms from our colleagues from bioinformatics. We take their algorithms and integrate them on such a platform. 45 minutes for alignment and variant calling, and then this analysis of the results with interactive exploration in real time. And the secret is all the data from the worldwide research centers are brought, are loaded, uploaded in the main memory. This is big enough. And then in the moment you have all this data in one place, you can immediately check whether there are some information available in one of this international research database. So overnight, the actualization is done, so the data are taken that are changed, and in this way, you can, as a physician, browse and get immediately information. What is nowadays difficult, and without this in-memory technology, it would also be difficult, because this is a question on memory. So here is the architecture of our high-performance in-memory genome system. On the basis, there are the data about the amount of data I have already spoken. Then there is a platform, this in-memory database. All the data are brought in from the different variants, from the mutations, all what is known. On the side, you see the uploader, so that this is really the actual data that are available on your research. And then on top of this, there are the different applications. So for example, a real-time analysis tool for the genome browser you have seen, or alignment coordinator, or others. So in-memory technology, I have no chance to go into much detail. It is a collection of many different things that were discovered during the last years. There is a partition idea how to, we use in the back, this was run on our HPI FutureSoc lab on a 1,000 core machine. So part of this task is to distribute, to participate the big task into smaller tasks that are assigned to the different processors. There is this multi-core parallelization, there is a compression, light-weighted compression. See this, no chance to go into details. This is a computer, and this is not a it is a supercomputer, of course, but it's not what you read as a supercomputer if you read newspapers. This is all the computers they try to make world championships in computing. This is something that's available tomorrow or a couple of years on any place. So this machine, it's one of the first, there exists 3 on the world, is 1.5 million. So this is compared to the supercomputer, nothing. So this is 40 cores. Each core has 1 terabyte main memory. So altogether there are 25 terabyte main memory. And this machine was used for the experiment we showed you. And what you can take home. Sequencing, we spoke about next generation sequencing, machines become faster. They become faster and smaller. Before I showed you a traditional, here's a recent one of this machine, and they generate more and more data. An immense data sets, and the problem is it comes in a very heterogeneous format, so that's it's difficult to compute. IT technology, therefore, is a key to explore and analyze this data because of this big data set. So all technologies that are available, that's parallelization, ideas of parallelization coming with this multi-core approach, that is high memory coming with this in-memory technology, they can reduce the time that's needed to analyze genome data. And this in-memory technology enables real-time analysis and interaction so that it becomes available, this technology, for treatment, not only for research. And I started with a 10 million prize, but we are more interested in the time. So here, let's provide technology that the identification of the genomic roots and the optimal treatment can be done before patient wakes up from anesthesia. So within 45 minutes. Thanks for that.
So I've been told that we are on a tight schedule, but I think, with such an interesting couple of presentations, there's maybe time for 1, perhaps 2 questions, depending on the length of the question. I've got a ton of questions. Yes, please.
The question for Dr. Bienig. Thank you very much for your talk. And I was wondering if you could elaborate on how can you identify few parasites in slides for people that, perhaps with some disease, you have a really hard time to find them in some tissues like bone marrow for visceral leishmaniasis in patients.
Yeah, you're right. Some of the structures are recognized by humans and very difficult to recognize by a machine. But if you sit together with a person and ask him how did he recognize those structures, you figure it out after a while. You certainly cannot implement it right away. It usually takes this interaction quite a while. You learn from the expert and you bring in the knowledge, and sometimes you find out that the knowledge of the expert is wrong. Yeah, he thinks I recognize it because of this and that, and he tests it out, it's completely fails. Yeah, so then you figure out how does he actually recognize the structure, and it's sometimes a little bit of a tedious process. It might take, in some cases, even some weeks. Yeah, if it's a very difficult task. But after a while, after going through very fast cycles of how do I learn from the expert, you can do it. I never stumbled over a problem where we could not implement it. Sometimes it takes a while. Sometimes goes really fast, but you always end up with a solution.
Okay, 1 more question.
Okay, I'm Linda from Nigeria, and I wish to congratulate you, Dr. Bienig, on the great innovations you are doing. With the concept of health for all and health equity, many of the innovations you've talked about here are really not accessible to low-income countries and rural places like maybe my school or my area. So, and this leads to a lot of guessing because, like, in my school, I have access to maybe light microscope, and there's a lot of guessing in diagnosis, and this leads to poor diagnosis, poor treatment, and very low life expectancy for the patient. My question is, or there are two. Are there innovations that are really cheaper and that were developed bearing in mind the low-income countries? If yes, I would really like to know. If no, is there any initiative to subsidize the cost of these machines for low-income countries? Thank you.
Yeah, that's certainly a very good question. I'm not sure whether I understood everything perfectly, but I think IT can certainly contribute a lot to countries with lower incomes, because I mean, machines are getting cheaper and cheaper, and also via the internet you can get service from all over the world. Yeah? And also, if machines does some analysis for you, you can learn from this. I mean, all the experts bring their knowledge into the machines, and the machines can then do a relatively good job. And that can be done used not just for therapy or diagnosis around the world, but it can also be used for training and education, which is, I think, also very important, so that you can distribute all the knowledge in a very fast fashion around the world. The knowledge which is usually today just located in rich countries. Yeah. So, did I answer your question, or maybe I...
I'm okay.
Yeah. Thank you very much. We're done. Wonderful. This is really interesting.